Hey, Vincent,
Hey, can you hear me okay?
I got you.
Okay, I'm gonna try to set you up as actually, if you, I'm letting everybody in and I'm gonna go ahead and start a quick presentation to introduce you. And then we'll set up the screen sharing for you. How's that?
Sounds good.
Good, okay, great. Hey, everybody. Sorry, can you hear me, bud?
Yeah, I got you.
Okay. Hey, everybody. Sorry I'm a little late. I had some trouble with Zoom trying to get in. So I'm a few minutes late. So I'm going to go ahead and start. Let me just get going here. Go off the desktop. And let's see, share screen. Where is that? Okay, new share. We're going to be on here. All right. And we should also be recording. Let me make sure we're recording. Yeah, we should be recording. So anyway, if everybody can see that. Can you see my screen, bud?
I got it.
Okay, yep. Great. Well, anyway, so again, apologies for being late, everybody. We'll get started right now. And welcome to our latest installment of the Martin County Water Ambassadors Program. Just so you know, we've got our whole lineup pretty much filled until September. And these are the list of webinar topics and speakers. So our next one will be on June 20th and presented by Ms. Kalita Scott, who is working for the Indian River Lagoon National Estuary Program. She's going to talk about her work in engaging the community on IRL issues. And I will put the Zoom registration links as we go in the chat during Mr. Howard's presentation. The other thing I wanted to remind people that are here with the Florida Nurseries Growers and Landscape Association, you are eligible for receiving 1 unit CEU for attending the webinar. And the information is there. You're probably, if you are an FNGLA member, you're probably familiar with Ms. Mary Mott. So you can contact her or contact me directly at the email, and I'll put my email in the chat as well. We are, aside from the lineup on the flyer, we do have a September 5th webinar planned as well. And that we're getting that topic and speaker finalized, but should be very soon. So Lisa Krimsky will send out an updated flyer related to that. And now, just so you know, if you've been looking for the recordings or wondering where they are, all the webinar recordings are now on our Martin County Extension YouTube channel. And again, I'll put that link in the chat. And but if you Google UF-IFAS Martin County Extension on YouTube, you'll find it. And we have all the 2023 webinars up to date. And we also will have past recordings there too. I'm sort of slowly adding them in, but they are organized by year. So look for the 2023 playlist for this year's recordings. And this one will probably get up today, or not today, but we'll get up on the YouTube channel probably about a week or so. So look for that. We are recording this webinar. And again, we're putting those on the YouTube channels. So please use the chat for asking questions. You can ask them anytime during the presentation, and we will get to them at the end of the presentation. Okay. With that, I'd like to introduce Mr. Bud Howard from the Loxahatchee River District. We're very happy to have the Loxahatchee River District. We have two, Mr. Howard, and then I think in August, or we'll show it again. The director, Dr. Aubrey Arrington, is going to give a presentation as well. And just so you know, the Loxahatchee River District is, parts of it are located in Martin County and Palm Beach County as well. So today's presentation is going to be given by Mr. Bud Howard. He is the Director of Information Services at the Loxahatchee River District. He has a master's degree in ecology from Florida International University, and he has an extensive experience working in the field. He also likes to do a lot of things with computers and data, and so he's kind of responsible for a lot of that. And he's currently leading the WildPine Laboratory at the Loxahatchee River District. If you've been there before, you may have been to Bush Wildlife, which is a great place to visit, by the way. And he is leading the laboratory, customer service, and their IT departments. So, Bud, I'm going to go ahead and stop sharing. And let me see if I can get you set up here and turn it over to you. All right. You are a co-host now, so you should be able to share your screen.
All right. Are you seeing my presentation?
It is coming up. There it is. I see it. Perfect.
Very good. Thanks to everybody for being patient. Nothing like some technology. When it works, it's great. When it doesn't, it's not so great.
Yeah.
So this presentation was originally supposed to be from Rachel Harris, our senior scientist, I should say, our former senior scientist. She since has left the district, excuse me, left the district to pursue a new career and be able to be closer to her fiance down in the Keys. And so we were very disappointed to lose her. But I wanted to kind of broaden out the topic a little bit today to talk more than just seagrass. I'm definitely, for those folks interested in that, we have some very interesting stuff to share with you as it relates to that. But I wanted to broaden out the scope a little bit and be able to talk with you about some of the other things that the Loxahatchee River District is involved with. Let's see here. Let me make sure we got all this running. There we go. Seems to be good. So forgive me. All right. Forgive me. The Loxahatchee River District, first one I'm going to do is we'll talk a little bit about the district, some of the things that we do, some of our program areas. Main topic of work that main area of work that we do is serving as the regional wastewater utility. So I think it's important to shed some light on that and how septic systems and sewer systems function in Florida, or at least in many parts of Florida. And then talk about the Loxahatchee River in general and some of the watershed aspects and river restoration work that's going on by many entities in the area. And then we'll get into some of the science of the neat work that we're doing and give you links at the end. We really try to make sure that we get as much of our information out there as possible. So hopefully you'll find some of that stuff valuable. So the Loxahatchee River District's an independent special district of the state. For those not familiar, special districts are established by the Florida legislature to fulfill certain missions or duties throughout the state. And ours is to protect the public health and help to preserve the Loxahatchee River. We'll get into some of the neat details. If you haven't been down here, we definitely encourage you to come see this beautiful treasure that's in our backyard. We fulfill this mission through three main work areas. One is serving as the wastewater utility. Each day we treat 6 to 7 million gallons of wastewater that comes from the community of Southern Palm Beach County and Northern Palm Beach County, Southern Martin County, and serving the communities of Jupiter, Tequesta, parts of Juneau. And again, 6 to 7 million gallons of water come to us each day. We treat that to a high level and then send it out to one of 9 area golf courses for reuse as reuse or irrigation quality water. And planned communities such as Abacoa, which is just off of Donald Ross Road, And even the ball fields at Roger Dean Stadium are watered with our irrigation quality water. So it's a great example of recycling a valuable resource in Florida. As I said, we do a lot of research and monitoring on the river, really trying to keep our finger on the pulse of the health of the river. And we also have been involved, that's how Vincent and I think first met, was involved in some neat restoration projects. We did some oyster reef restoration projects a number of years ago, and are very involved in all aspects of the river. We have an environmental education program through the River Center. If you haven't been there, it's at Burt Reynolds Park in Jupiter, and a neat facility that kind of takes you through a virtual tour of the river and really helps to educate the public on water resources. And they do a lot of neat activities of getting out and experience the watershed. All right. I'd be remiss if I didn't talk a little bit about wastewater and some of the things that we do. Most of us don't think much about water as long as it leaves our houses and our businesses. We often don't give much thought to it. But as we've seen in the news lately, there's a couple different ways to get rid of wastewater once it leaves your house or your business. And one of them is through septic systems. And the water leaves the house, goes into a tank where the solids settle out, and then the water overflows into what's called a drain field. And that area is home to a whole myriad of different microbes in this area that's dry, or it's also called a vadose zone. And so this dry area is really critical to house the microbes that break down the nutrients and other things that are in that wastewater. So the problem is that in Florida, as you know, particularly during the wet season, this water table can be very high, and you just don't get good treatment when all of that water is swimming in the same water as what's out in your roadside ditch. And the microbes just don't thrive in the wet environment. And furthermore, all this water is moving towards the nearest larger body of water. And so a great example of this is we can actually model wastewater streams coming out of septic systems. Here we're looking at an area in the Loxahatchee River referred to as Pennock Point. And each of these little dots represents a plume of pollution coming off of a septic system. And this is all based on soil types and elevations. But you see how that water quickly leaves that property and starts its journey over to the nearest water body. And so particularly in the dry season, when that septic system and the roadside ditch and everything is all swimming in the same water, it's all running to the nearest water body with little to no treatment. And so that's why septic systems are a hot topic now. And many, again, in many parts of Florida, particularly those low-lying, they're just not very effective. So a better way to do that is through the centralized sewer system. So water leaves your house, comes out through a network of pipes into a whole network of larger pipes and pump stations that get that water to a water treatment facility like ours here. And you treat that water to a high level. And then in our case, we go ahead and take it to the next level where we're actually recycling much of that water, particularly during the dry season when water's needed most. So it's a big undertaking to do this. And an example here is, just a snapshot of many of the properties that were older properties on septic systems that we converted to central sewer. And then in addition to this conversion program, all new construction was brought on to our central sewer. So as of today, this line represents I-95 and the Turnpike. 99% of all the properties east of I-95 and the Turnpike are on our central sewer system. There's only a few remnant properties out here that we're still working to get converted. So it's a major accomplishment and a real shout out to the legacy of leadership in our community over 50 years ago. So much discussion now is about how septic systems are not, the best solution in Florida. And here, the folks 50 years ago in 1972 got funding from EPA to develop a pollution control project and facilities in the early stages of our wastewater treatment plant. And so this area down in here on the southern tip of Jupiter Island, referred to as Jupiter Inlet Colony, we modeled all the septic effluent coming out of each of those properties. And all that now is deleted because those properties are on the central sewer system. So it's a good thing for sure. When you realize that, when you start to appreciate the magnitude of pollution coming out of those systems. All right, so let's shift focus a little bit over to the Loxahatchee River. For those not familiar, Jupiter Inlet, we have Jupiter, the community of Jupiter here, Jupiter Inlet, the southern Indian River Lagoon through this section. The Intracoastal Waterway goes off to the south, and then the main fork of the river is the Northwest Fork. These numbers represent mile markers. If you were traveling by canoe or boat, this is about 14, almost 15 miles to get all the way back to Indiantown Road, which is here. You may be familiar with Riverbend County Park, Palm Beach County's park here. Great place to go and rent a kayak and go travel down the river. And I love to travel from this section of Riverbend Park down to the second salt dam. There's these small dam features out there to Maston Dam and then paddle back. It is truly a magnificent part of the river. There's also the Southwest Fork. There's the flood control structure operated by Water Management District here called S46, and then the North Fork, which goes up into Jonathan Dickinson State Park. So again, I mentioned this area back here in the upper, to take you on the upper reaches of the river, take you on a little virtual tour of this part. It is truly magnificent. If you haven't experienced it, this is a great time of year to go. The cypress trees have all popped and are full and provide lots of shade, and the river's not flowing too hard to paddle back upstream. And it's a magical, beautiful place. And to think that this was a couple signatures away from becoming housing developments and strip malls and everything else, we're fortunate to have this in our backyard. As you continue downstream, you transition from that freshwater cypress habitat into more of a mangrove dominated system, and you start to see some oyster reefs and things like that, where the salt water that's coming up through Jupiter Inlet is colliding with the fresh water coming down the river. And then further downstream, kind of at the terminus of the river, we're in the heart of the estuary portion, and we got Jupiter Inlet here. And this lower estuary is dominated by marine water that comes and goes through Jupiter Inlet. And so this is where we see a lot of the seagrass and various other things, as well as up and down the Intracoastal Waterway. The Loxahatchee is really fortunate in the sense that there's been a whole slew of different agencies that have worked hard to acquire lands through various programs to protect the habitat, which then provides the waters that feed a lot of the Loxahatchee River. Down to the south, we have the city of West Palm Beach has Grassy Waters Preserve. It serves as much of the city's drinking water supply, but it's also a beautiful, pristine area. Palm Beach County has Loxahatchee Slough Natural Area, Corbett Wildlife, Hungry Land, a bunch of huge parcels and properties out to the west. And then Martin County has been instrumental working with Palm Beach County to do amazing restoration work in Cypress Creek area. And then off to the north, we got Jonathan Dickinson State Park. So it's, we're fortunate in the sense that we have so many natural areas and so many areas that were protected. All of these agencies have been masters at leveraging, the land acquisition and protection and restoration work. It's our water quality and our quality of life definitely benefits from this. So if you haven't explored these areas, I encourage you to do so. They're beautiful. So the river really is dealing with competing forces depending on the time of year. And so during the winter and spring, when we're dealing with very little rainfall like we have been recently, very little water flow is coming down the river. Marine water's coming up the river, and what's happening is over the years, there's been clear evidence of harm to those freshwater cypress habitats up in this upper reach of the river. And so that's one of the main reasons behind huge efforts to provide additional flow to the river so that we can kind of push that salt wedge further downstream and try to provide protection to those beautiful habitats. Now, in the summertime, when we get into the heart of the wet season, the pendulum swings to the other extreme where we're all drowning in water, flood control, and water managers are working hard to make sure our property doesn't flood, and huge amounts of water gets shunted into the river system. And then that can have a profound effect on the lower estuary that are used to spending most of the year in more of a marine habitat. And so when you start to look at some old aerial photographs, I think it really helps you appreciate, the profound changes that we've all had on this. It's tempting to want to point fingers at one industry or the other, but the fact of the matter is all 22 million of us are here, and we've had a profound impact on the whole ecosystem. So what can you do about this? We're all here. What are our best opportunities? Well, as I mentioned, there's a lot of work going on between particularly the Water Management District and the Army Corps of Engineers to try to get that little bit of extra water to the river during the peak of the dry season. Our executive director, who you'll hear from soon, he has a great expression. The river needs a thimble full of water, but it needs it at the time that everybody is clamoring for water. So it's a challenging undertaking. And then trying to manage the discharges the best we can during the summer season. Tip of the hat to the Water Management District's operators. They've been able to hold back water, in amazing ways during the peak of the wet season when the whole area is saturated. And I think that's another testament to those natural areas to be able to store that water in there. Make no mistake, and we've been through kind of a period of heavier than normal rain. And there is no bucket big enough to capture all the rain to prevent all discharges. But again, that's where doing what you can with what you can put together is how you actually make some progress. The natural areas are a key part of all that water storage and being able to use that water during the wet season, or the dry season rather. Pollution management, I think this is a big one. Clearly septic tanks are a big part of this when you look at the nitrogen inputs from those, as well as other things. Stormwater, this one I think is such a big area of opportunity. Many of the older communities that were built before there were more stringent stormwater requirements, basically their objective was to get that water to the nearest biggest water body and get rid of that water as quickly as possible. My neighborhood, for example, was built in the 60s, and you can shout through the storm drain and hear it echo over across the river the next block over. So I think there's opportunities, to try to treat some of that water and figure out ways to keep harmful things from going into the river, as they have done through best management practice and the construction industry in large part, in agriculture and other areas where things are very different than they were decades ago. And so I think there's been real progress made there. And then through habitat restoration, again, tip of the hat to all the agencies that have been able to do amazing things in all the different natural areas. And I think it's important to remember that it's worthwhile to try things. If it's not working perfectly, maybe you can adjust it a little bit, but try things and adaptively manage as you go along. And so I keep mentioning trying to get that extra water to the river, particularly during the dry season, to prevent that harm to the freshwater cypress habitat. The Corps and the Water Management District have been working for many years now. And, finally now I feel like we're making some real progress. Congress has authorized the project to actually do this work and to try to get water through various channels to the river during the times that it needs it most. Another area that many agencies are working together to help protect the river is through the pollution reduction plan. DEP, under the umbrella of EPA, assesses impairments to water quality. We have identified impairments in the Loxahatchee River. It's a watershed approach, so it's not just little corners of the watershed. It's looking at the whole thing, and all agencies are working together through an organized, coordinated group called the Loxahatchee River Management Coordinating Council. And so it's neat in the fact that the legislature has set up this group and really helps to drive the efforts underway to protect the river. So you've got our agency, Town of Jupiter, Village of Tequesta, Park Service, Water Management, DEP, DOT, Jupiter Inlet District, and many others. So all at the table working to try to make improvements to the river. And more recently, we've done this through the development of this pollution reduction plan. And very clearly, we're laying out a whole series of water quality improvement projects to try to get us out of those impairments. So now we'll shift down a little bit more into the weeds with some of the environmental work that we're doing. We have several water quality programs that we work on. One of them is the weekly bacteria testing that we do in areas that are popular for recreation. This is looking at enteric bacteria primarily in the lower estuary. And our thought process with this is, how do we get information to the community that helps them kind of make a decision on a given weekend? Are we going to go play in the river, or is this maybe not the best weekend to do it? Because let's say, for example, it's been a period where we didn't have much rainfall, and now we got a big rain and everything's washed off our streets and yards, and bacteria counts are high in the river. We also have a volunteer program that goes out and takes a look at several key parameters on a weekly basis. We have our comprehensive monitoring through our River Keeper program. We have several sites that we monitor monthly, and then another suite of sites that we look at throughout the watershed on a quarterly basis. And we're looking at a whole slew of parameters there, the total nitrogens, the total phosphorus, and many, many parameters. And we also do this work in partnership with JD State Park and Town of Jupiter and some others that help with the data collection. Lastly, on the water quality side, we have some instrumentation that we put out in the river that looks at salinity and temperature primarily on a very frequent basis. This is really interesting to be able to see those changes as, in particular salinity, I think is interesting. I'll take you through an example of that as we shift from an outgoing tide that's dominated by fresh water to an incoming tide that has the marine water. We'll pile into some of the seagrass work that we're doing. We also have been involved in looking at oyster spawning activity, mapping the oysters throughout the watershed, and some of the restoration. Of course, we're not doing all this independently. There's other agencies doing work here also. So our weekly bacteria map is one that we're proud of. Each week we go out and sample, again, areas that we think are largely used for recreation purposes, with people that are either water skiing, having fun in the river. That we want the community to kind of understand what the basic condition is, this as it relates to bacteria. And this is based on the Florida Beach Watch program, and so it's a very restrictive criteria, and those dots turn red when it doesn't meet the criteria. And by and large, you can see a pretty strong correlation to, particularly the example I gave you, where when it hasn't rained for a period of time, we get a big rainfall, you'll see some of these numbers jump up. The rest of that story is that the good part is that we know that we don't have septic effluent pouring into the river, which I think is a big relief. For our River Keeper program, we make a bunch of different maps available. I like to call these our stoplight maps. We've got monthly maps that summarize the monthly data. We got annual maps. And then this one, which kind of gives you a pie chart of the proportion of samples for the years selected. In this case, total phosphorus, we've got total nitrogen, and we've got chlorophyll. So the proportion of samples meeting the water quality criteria established by DEP and EPA for that location. And so each location may have different criteria, but this is the proportion of samples that meet or fail to meet that criteria. And so you can see we do pretty well on the nitrogen side. The total phosphorus side, we see a little bit more color, particularly in some of the tributaries, but sometimes those aren't meeting the criteria. And then on the chlorophyll side, which is interesting, we do see a lot more color. And it's important to note, though, that part of the modeling that went into establishing the water quality criteria, I think, struggled with the volume of data that we had. And so some of these water quality criteria are incredibly low in, say, for example, the single digits. Well, there's many folks that, manage watersheds that would, cut off a limb, I think, to be able to have a chlorophyll concentration of 3. And here we're failing to meet, in some of these areas, we're failing to meet the water quality criteria if we're above 3 or 5 or some of these different numbers. So we take these a bit with a grain of salt, but it is an interesting situation where we don't have obvious nitrogen or phosphorus issues, but we also have some instances, and sometimes they are genuinely high numbers. The old criteria for chlorophyll was like 20 micrograms per liter. And sometimes more recently, particularly when we don't have a lot of flow in the river and we have warm temperatures, we will see some really high numbers in there, like we have over the last month. So I hope, if there's one thing I hope you take from my presentation today is this slide, right? We're looking off to the east here. We got Jupiter Inlet up at the top here. And this is a great photo that kind of captures the 2 dynamic forces, if you will, of marine water coming through the beautiful, flat, clear day. Marine water coming into the estuary, and then the tides have just shifted, and now we have that fresh water coming back down through the system. And so you really see that swing between these two. And often people are like, why is the river so dirty, and this is so bad, it looks so awful, and this and that and the next thing. But the message I hope you'll take from our presentation today is that brown water is not always polluted water. Again, I think we benefit from the enormous amount of natural areas that we have. And so what this water is it's stained with leaves that have it's fresh water that is stained from tannins that are released from the leaves as those leaves decompose in the water. And so it doesn't necessarily mean it's polluted. And in fact, we've got mountains of data that show that this water is incredibly clean and safe. It's just that it's fresh water that's tannin stained, and it's heading out through the inlet during the outgoing tide. For the instrumentation side of things, I wanted to take you through a quick example down here, and all this information is on our website. You have Jupiter Inlet down here. As you're moving up into the river system, you come to our North Bay site, here, indicated by the black dot here. And this is an example of the instrumentation data for salinity, looking at 15-minute observations. So you can see each tidal cycle. The incoming tide drives the salinity up, and the outgoing tide drives the salinity down. And you can see that we're about 35, 36, 37 parts per thousand, or PSU. And on an outgoing tide, after we've maybe had some rain, maybe that's dipping down into the mid or upper 20s. So if you're an organism such as seagrass or fighting conch, or one of those neat things that we see in the river, you're making a pretty good living in the saltwater. If you move upwards, up into the Northwest Fork a little bit, you come to our oyster reef site. And pretty remarkable in the sense that salinity here is swinging from single digits below 5 all the way up to over 30 on a given tide cycle, right? In 6 hours, you're going from one extreme to the other. And I think it's pretty remarkable that organisms thrive in this pretty harsh swing in salinity. And then lastly, if we move all the way up to kind of that boundary where we start to transition out of mangroves into the freshwater cypress habitat, maybe we'll see a little bit of a salt push during low flow situations. And then maybe we get some rain and it'll go pure fresh through the entire tide cycle. And then as river flows diminish a little bit, you'll see little bumps in the salinity as it pushes its way out, up from Jupiter Inlet all the way up into that section of the river. So I thought that's a great example of kind of seeing these different habitats and how they vary from a salinity perspective. All right, so piling into seagrass. Obviously, seagrasses are important. Their food. Each of these photos, I think, was taken by our staff. And, you see foraging green turtles, manatees, sea hares, all kinds of neat things. So habitat's obviously important, sediment stabilization, carbon sources or sinks and sources. It's a neat habitat. I think it's important, though, to realize that seagrasses in the Loxahatchee River and many parts of the Indian River Lagoon are not these beautiful, lush, turtle grass beds that you see in the Florida Keys or in other parts. They're more diminutive, kind of sparse, but nonetheless, I think they're important and a valuable habitat. The Southern Indian River Lagoon has been a really interesting kind of reference site for us. We had done some work in the past looking at those areas, and some beautiful seagrass up there, but also have shown some declines, unfortunately. So our two programs are what we call our patch scale monitoring, where again, Jupiter Inlet down here, we try to look at a series of locations kind of in an upstream to downstream run across the river. We also have done some work up at our Hope Sound site, which is kind of like a reference site. And interestingly, this site is not influenced by freshwater inputs. It just doesn't. The salinity in this section of the in the Indian River Lagoon stays quite high, and you don't see the brown water that you would see coming down through the main parts of the river. And so we're going to these sites, we're deploying a bunch of 1 meter quadrats several times throughout the growing season for seagrasses, and we're looking at the percent occurrence. Our other program that we do every several years is, I call it a shotgun blast of all the potential areas for seagrass, to kind of look at the extents and then the change in seagrass at those locations. And so we've done that we use a great big 9 meter quadrat that we affectionately call Quadzilla, to be able to kind of take a big snapshot of a larger portion of the river floor. And we've done that over the years that you see there. And on the patch scale monitoring, if we consolidate all the data from these sites, from this the upstream to downstream part of the river, you can see that, and average all that data together, we can see that unfortunately we've been on kind of a bad trajectory here. Again, the data just tends to be disappointing, although we have seen some improvements over the past few years. But if we break this up by the individual sites, right, we've got our inlet site here, and then that's the black bar, and things look pretty good there. In contrast to our Northwest Fork site, which is this one here, which is the yellow line, following the storms some of the hurricanes that we had, those areas just haven't declined. Interestingly, though, this timing corresponds to, I think, if you look at the historical rainfall record, a period of time of more fresh water coming into the river system. And so some sites are doing better than others. We continue to monitor it and see how it looks. On the landscape scale, we look at things with painting with a broader brush to kind of look into the upper extents of the river, kind of see how things look at those locations, as well as down in the central embayment. And so you can see that between the data between 2007 and 22, quite a few changes, and particularly in the upper reaches of the river, right? You just don't see that grass showing up like we used to. And you also see some types of change when you start to look at it at the species level in the lower estuary. So what's going on here? We see these declines, and you read about them in scientific journals, in the newspaper. Indian River Lagoon, other areas are experiencing lots and lots of seagrass loss. The International Union of Conservation and International Union of Conservation and Nature, IUCN, did a report a number of years ago indicating that their calculations showed about a 7% decline in seagrasses globally. And what's interesting is if you annualize our data and look at it in a similar fashion, it's right at 7%. So it's very disappointing in the sense that we just kind of see this stair step decline. So what factors do we think are at play here? The Loxahatchee offers kind of a little different perspective, and the IRL South offers a little bit different perspective as it relates to the issues, the common issues that are referenced with regard to pollution and nitrogen pollution and various other nutrients. we don't have any, we have a very low residence time. A molecule of water up at Riverbend Park is out through the inlet in less than a day in the Loxahatchee River. So we have a low residence time, lots of flushing occurring through Jupiter Inlet. And then we also don't seem to have any profound nutrient problems. So it's a little bit different situation in relative comparison to other water bodies around the state. One of the things that I think is important to note is that this lower estuary is subject to that outgoing tide and lots of fresh water in that outgoing tide. But that Hobe Sound site, that water will stay clear, for weeks and weeks at a time, and you just don't see this freshwater input. So it offers a really neat contrast. So the factors, and this is my personal list from nearly 30 years of playing around in these waters, both recreationally and for work, is I think water clarity and light attenuation is a real factor. We're doing some really interesting work. Jerry Metz of our office is doing some really neat work looking at using light sensors positioned on a pole to be able to look at the change in light as it goes down into the water column. Because you'll have situations where this fresh water will ride on top of the salt water. And so you can see that, the light may not penetrate through that dark water as well as it does the clear water, but you can start to see how that changes and that light diminishes, and there's meaningful reductions in that light. Likewise, I feel like this is becoming a two-part hit for changes in light attenuation and, in this case, sedimentation. It's a double whammy. We all want to have nice beaches to enjoy. Palm Beach County and the Army Corps of Engineers have done huge beach nourishment projects, but I feel like there's a meaningful fraction of fine sediments that are coming up on the beach with those projects. And you see that plume of fine sediments coming into the inlet, and it becomes trapped in the waterways. And so not only is that reducing light, but I think you're also providing a source of sediment changes, and that's where we get into the accumulation of sand, particularly in the lower estuary. We're seeing changes occurring there. And so likewise with the outgoing tide, you're seeing a salinity flux. During periods of high freshwater discharges, that salinity will go down for a meaningful period of time, which is why we're always working with the Water Management District to try to minimize those harmful discharges, and when they must get rid of water, to do so it doesn't overwhelm the salt water coming in through the inlet. Likewise, we've had some record temperatures, and we see that in our water quality data. And organics, I think, is another big one, right? How many times do we see folks, blowing their grass clippings into the nearest storm drain? That storm drain is flowing directly to the river, and you see that in the river when you're snorkeling around and doing work. There's areas with just massive accumulations of organic rich sediments. Likewise, we also see damage and scour occurring. I suppose I'm aging myself, but it wasn't that long ago, it seems like, that a 25-foot Mako was a really big boat. Nowadays, that's a small skiff, and that people are tearing around in 30 and 40, 50-foot, big center consoles. And the wake that goes with those boats also does cause visible damage. And so it's important, I think, to help the boating communities appreciate what kind of damage that can occur. And then, of course, we see the damage from storms. One of them that you don't hear much about, but I definitely, especially when you think about the magnitude of change in seagrasses all throughout the Indian River Lagoon and other areas, is I wonder if there's some disease lurking that we just haven't identified yet. A couple things that we have done in our lab that's really interesting that help us kind of rule out some things has to do with, one is the Roundup or glyphosate herbicide. This has been in the news a lot of, health concerns as it relates to, the astronomical amount of use of glyphosate in agriculture, but also too, I'm sure the home improvement stores sell a heck of a lot of this stuff for household use. And so it was interesting. Lorene Bachman was our former lab manager, and she always had a suspicion of, I wonder what effect this is having, because we know that there's, big properties all, adjacent and near the water in the Loxahatchee River, where everything that runs off our yards and streets is going straight to the river. And as well as use of these types of products and properties that are right up adjacent to the river. And so Rachel, who's supposed to give your presentation today, but had left and partnered with a great student from Florida Atlantic Honors College and Owen Severa, and they set up a really ambitious project in our lab to try to evaluate some of this. And so they established 12 tanks with seagrasses in them and broke it into basically 3 different treatments. They had an area, one of the treatments, they took the water down to simulate a low tide situation and just left it and refilled it, and that was the control tank. The second treatment they did was take the water down to simulate a low tide event, and then actually sprayed it with Roundup as you would if you were treating weeds around your house, and then filled it up with clean salt water and continued the study. And then the last one was to try to simulate maybe a spill event, if you will, where they took the water down to simulate the low tide event, but then filled it with pretty concentrated Roundup in the seawater. And I can tell you that the results of this were not certainly what I expected. And so let me take you through some of these figures. We got some nice kind of pictures here that kind of show, how things went. We're looking at 2 different seagrasses, Alophila ovalis, which is the formerly Johnson seagrass, which has now been renamed. I'll probably refer to it incorrectly as I'm speaking. And then Halidulu redii, which is shoal grass. And we're looking at the shoots per tank. And if you're not familiar with these are box and whisker plots. These basically plot the distribution of data among those 4 tanks, right? So on the red ones, that's the high dose, right? So we took the tanks down, in this case, filled that tank up with fairly concentrated Roundup, it mixed in with the seawater. And over the span of the days, which is on the x-axis here, they were going along the seagrass were going along pretty well. And at about day 5, things started to decline rapidly. And in fact, most of those seagrass in those tanks, or essentially all the seagrass in those tanks, died. And that's represented in this lower figure here. That's for the formerly Johnson's grass, ovalis. For shoal grass, there's less density, so fewer shoots per tank, but same deal, went along, was doing okay. About day 7 on those, they kind of quickly fell off the cliff and perished. Now the low dose, again, this is the one where they sprayed the seagrasses. Surprising thing happened, right? The seagrasses were going okay. They started to decline a bit, but then they actually came back and survived, which baffled me. And if you have a small closed system here, a little 10-gallon aquarium, I thought once you put some Roundup in there that they'd be gone. And then lastly, the clear bars or the white bars are the control tanks, and you can see that those fared pretty well throughout the experiment. So again, the low dose, again, the gray bars, right, sprayed those seagrasses directly, and they've surprisingly fared okay for the shoal grass also. So not what I expected, and that's why you do science, right? The interesting thing is this glyphosate can stick around for a long time. We tested it going out past 53 days, and it was still very present in the water. Curiously, the overspray from the Roundup doesn't seem to be a real hard hit to seagrasses, but it does beg the question of when you start to mix together stressors, do some of these things maybe start to play into it. Switching over to seagrass wasting disease. Rachel was instrumental in getting some work done with this. She partnered up with some of the professors at University of North Florida. Labyrinthula is the name of this slime mold. And this pathogenic protist, recall if protist is not an animal, not a plant, not a fungus, one of these kind of weird organisms. But it's some of these Labyrinthula species are pathogenic to seagrass. And what I didn't appreciate is that, work had been done in the 1930s that illustrated that this 90% of the eelgrass loss in the North Atlantic during this massive die-off in the 1930s was attributed to the species of Labyrinthula. So again, some folks at University of North Florida are doing some neat work in Florida Bay. And what they're finding in essence is that in Florida Bay and areas that experience stressors with either hypersaline or various other stressors, that the Labyrinthula and the pathogenic version of that is actually causing some of this seagrass wasting disease. And they see higher evidence of the pathogen there. So the curiosity, to Rachel's credit, was, wonder if, we have some of that going on here. And so they went and collected seagrasses at a couple different locations. Our North Bay location, just west of the railroad tracks, and then up in Coral Cove, got a variety of different species and sent those off to see what they found from a culturing standpoint. And then another phase, once they got through the culture phase, to identify which one. So again, phase one was the culture and microscopy. Do we have it on our seagrasses? And it turned out that in fact we did. The prevalence ranged from 38 to 48% by the site. The Coral Cove site wasn't meaningfully different than the North Bay site. Both showed quite a bit of presence. And then if you break it out by species, the shoal grass had a lower prevalence than the more meaty type of larger bladed turtle grass. And so the Syringodium or manatee grass also showed quite a bit of this seagrass wasting disease. So that really got us interested and curious whether or not maybe this is what's driving some of the declines we're seeing in our area. So we kind of moved to the next phase, which was the genetic testing to identify which species of Labyrinthula these were. And curiously, out of the 13 samples they sampled, none of them came back as the pathogenic or at least the species of Labyrinthula that he knew were pathogenic to seagrass. So one of those interesting situations where it's, you look for a smoking gun, but we didn't find it. So I want to shift a little bit to some neat work that we're doing on the wastewater side with regard to wastewater surveillance. Whenever we use the bathroom, we excrete lots of virus fragments of things, and this all got started with COVID. And we worked with a company named BioBot early in the pandemic. And then later in the pandemic, Verily, which is the life sciences subsidiary of Alphabet, which is formerly known as Google, they are partnered with some scientists from Stanford University and Emory University, and they've broadened the scope of the testing in the wastewater. So we were focused primarily on COVID, and again, on our website, we've got more information as it relates to the different the whole time series of COVID. But with the Verily group, we've expanded to testing influenza A and B. We had a very early season of influenza A that thankfully kind of petered out early, and there was some concern among the scientists, would there be a second wave? We're looking at some respiratory viruses and noroviruses. If you have a small child, that norovirus can be a real problem. And so we're doing this work 3 days a week. We sample our wastewater, and this is going to be an evolving project where they're looking at other markers and viruses in our wastewater, which is in our community. Again, it's important to realize I think we all kind of discount COVID, but it's still very prominent in our community. Not nearly at the concentrations that it was, but it's still there. So lastly, what can you do, right? I think, my two big ones are, we got to advocate and educate. When you see, somebody blowing leaves and grass clippings into the storm drain, I think it's important to let them know that, that's not the best place to do that. And likewise, working with our local government leaders to help educate not just the residents, but also the workers. And that needs to be, a whole comprehensive education program so that people understand. I don't think many people maliciously do stuff to harm the environment, but being able to help them understand that picking up dog waste is actually important, particularly in those properties there that are in closer proximity to the river, because it's probably an older community and that pipe is running straight to the river. There's been some great work on various communities as it relates to fertilizer ordinances. I think that's a big one in keeping the nutrients down. Responsible boating, right? Anchoring, running your boat off of an area that you didn't even realize had seagrass on it, using the motor. Those are all things and opportunities for educating people. So there's my contact information. Please do not hesitate to reach out to me if you have any questions about any of the things I talked about. The wastewater surveillance is on our homepage at loxahatcheeriver.org under the wastewater surveillance. We have a big section here under protecting the river that has links to all of our water quality data. We've built some really neat tools that allow you to slice and dice our data a million different ways if that's your area of interest, as well as provide our seagrass data and various other things. Under this maps and apps, one of the fun things we put together is kind of an interactive map that takes you through all the different kind of a virtual tour of all the different resources and natural areas and parks and various other things throughout our community. So it's kind of a fun resource to go. Many of us don't even realize that, there's these beautiful natural areas right in our backyard and fun to go visit, and beautiful walking trails and other things. So with that, I'll leave it at that. And I appreciate your time. And thanks for letting us come talk with you.
Cool. Thank you so much, Bud. That was awesome. So we're open for any questions. While we're doing that, I'd like to launch a quick poll. It'll just take a couple minutes to do, so please fill that out if you can. So Bud, is there any idea of how much glyphosate might be in the environment, like in the sediments or even the water column in the Loxahatchee River? Is that something that's been looked at?
Good question. So we haven't done extensive testing on it, but we have done some spot checks here and there. A lot of that boils down to the detection limits in the lab, and they have some pretty amazing capabilities, but at this point they're not, we're not detecting it in anything noticeable in the waters. It's curious in the sense that I think dilution is the solution for some, and I think that's a classic example of it.
Right. And I just did a quick look to find out what the half-life is of and it's kind of all over. It's either a few days or 90 days. It seems like it's extremely variable. So that's a difficult one to pin. I can see that being a difficult one to pin down. So cool. Let me see if we got any other questions here. I posted the paper on Labyrinth.
That's a mouthful, right?
I know I can't pronounce it. So that's up there, and then of course the website for the Loxahatchee River District. The other thing I was going to ask you, Bud, is what are the opportunities for it? Are there opportunities for the public to be involved in some of the scientific efforts that the Loxahatchee River District does?
So there are a couple ways. One of them is that we have our volunteer water quality monitoring program. So that's a way to be, directly involved with the work that we're doing week to week. The other one is that if you have an interest in seeing some of this stuff firsthand, we do have on occasion can bring out visitors. And so We don't do big organized events, but if you have an interest in this, please reach out to me, and we're happy to bring you out on a field trip one day. On some of those big seagrass monitoring years where we're going out doing thousands of samples, we do try to enlist the help of a group of volunteers to kind of stick with us. One of the things that we kind of did in our debrief of this last year is, could we formulate a team of people that could help us knock out more work in a shorter period of time? So if you have an interest in doing some seagrass work in a couple summers, that's one that we'd love to enlist your help, and it's kind of fun work to do, and I think many hands make light work, and so it's one of those that we think that if we can establish a group of volunteers that can kind of commit to it, go through the training, but then also be able to commit to several days of work, that we could probably get a lot done in a short period of time.
Yeah, that sounds amazing. And just to add to that, but and maybe we can talk about this later, Florida Sea Grant has a citizen science seagrass monitoring program called Eyes on Seagrass, and maybe that's where we can kind of find a fit there and help out.
That would be fun. That would be great.
Cool. Oh, so you got some nice comments here that I'm seeing. There is a question or request from Bill Klein. What he wrote is, can you give me a good write-up about your water quality program that I could use for the IRL?
So as far as having a write-up, we do have, I encourage you to reach out to me, Bill. We have a variety of different things. We've got some synthesis of several decades of data and reports that are on our webpage under that Protecting the River section. If you go under the River Keeper section, there's a series of reports there. And also we provide a whole myriad of data visualization and summary tools on our website. So if there's particular sites that you're interested in, there's multiple screens of summary statistics and analysis and all kinds of neat stuff that you can use. And if you don't have something, or you can't readily make the visualization or the data summary that you're looking for, I'm happy to help. Again, we do this work to serve not just the scientific community, the public, everybody, right? So I'm happy to work with you on that, try to get that to you, as well as our full data sets on our website, so you can pull down that information and filter it for whatever areas you're interested in.
Yeah, those data visualizations are really cool, and I think that speaks a lot to your expertise, Bud. Yeah, no, that's really cool. I can get lost in that.
It's fun, right?
Yeah. All right. Well, I realized I was a few minutes late. Again, my apologies for that. And but I think I'm not seeing any more questions. There's a comment here. We are also broadcasting the webinars into the Blake Library in Stewart every month until the end of our year, so to speak, which will be in September. So we'll post that flyer again in our next webinar on June 20th, and we'll have some more updated information on our September 5th webinar. And with that, I'll let you go. Thank you so much, and thanks for your patience. And
it's all good.
Really appreciate it. And thanks everyone for coming. That was fantastic. All right. And I'm going to go ahead and end the poll, and I think we're good.
All right. Thanks very much.
Yeah. Thank you, Bud.
All right. Take care.
You too. Thanks.